Hello and welcome. I wanted to take a few minutes here to do a video on how to solve a Punnett square. Again, normally I would be teaching this in the classroom setting and I could help answer your questions, but there were a few students who felt like they needed some extra help with how to solve Punnett squares. So what I would like for you to do is I would like for you to watch the video and take notes, especially making sure that you have down the 4 rules on how to solve a Punnett square. Using your bill on page 63. And then as of always, if you have extra questions or if there's something that you didn't quite understand, make sure you ask. Okay, so let's get started. How to solve a Punnett square. The first thing I wanted to do was just go over some basic vocabulary that hopefully you were able to get from the textbook and from the videos that I had you watch. Big 2 vocabulary, phenotype and genotype. So phenotype is the physical trait of an organism. So if I look here, there are 3 moths on the screen, and they have a physical trait of color. So 2 of the 3 are brown, 1 of them is white. But a phenotype is that color or that physical trait of what you see, what you observe. The genotype, though, are the alleles that the organism has for the trait. So the alleles are noted by the letters here next to the moths. So the dominant alleles have the capital letter and the recessive alleles have that lowercase letter. So the genotype then is represented by the letters, and the letters represent the alleles. So another vocabulary term then that hopefully you know, if we have 2 dominant alleles, we can call this homozygous dominant. So homozygous meaning the same alleles. You can also have homozygous recessive. So here we have 2 lowercase alleles. So therefore they're going to be the 2 recessive alleles. And then if you have 1 allele dominant and 1 allele recessive, then they're 2 different alleles. So this genotype would be considered heterozygous. And you could also say that the organism is a hybrid because they must have gotten 2 different alleles, and each parent would have given them a different allele. So the 4 basic rules to solve a Punnett square. The first thing that you should do is that you should find the genotypes of the parents. So sometimes it might be given to you in the actual problem, and it might say a heterozygous is mated with a homozygous recessive, and then it's given to you. Sometimes that also might just be like they're giving you the phenotype. So if they're telling you that they have the recessive phenotype, like in this example here, if they were to say there are 2 white moths or light colored moths, then you could imply that they have to be this genotype of homozygous recessive. But that's the big thing that you want to do is you want to first figure out what are the genotypes of those parents. Once you figure out the genotypes of the parents, then you add them to the Punnett square. So put one parent's genes at the top, but make sure that you separate them over top of each of the boxes, because what will happen is when meiosis happens, those alleles will separate from one another during prophase 1. So we symbolize that then, but just by separating those alleles and putting them at the top, and then putting the other parent's alleles separated on it should say the side, not the bottom. And then you're going to fill in the boxes. So you're going to go down and then you're going to go left to right. So you're going to add those alleles to the boxes, and I'll show you an example of that in a second. And then make sure you answer the problem. So you want to make sure that you're answering what the question is asking you to. So you have to go back, look at the model you just created, and then be able to answer that problem. Okay, so for example, let's say I have this population of moths, and they have different genotypes and they have different phenotypes. So let's say I'm going to use this moth as one parent and this moth as the other parent. So the genotypes in this case are given to me, so I know what they are. It's going to be easy for me to find the genotypes of the parents. So then I need to add the genotypes to the Punnett square. Again, it should say top and side, not top and bottom. So I put one parent's genotypes at the top. So I put the heterozygous genotypes at the top. And then I put the homozygous recessive genotypes on the side, at the bottom, the side. Next, then I fill in the boxes. So the alleles from the top come down, and the alleles from the left come over. So I'm just going to fill in the box, each one of these. And then I need to answer my problem. Now, there is actually no problem here, but let's say I were to ask a question as to how many of the offspring would be light-colored moths. Then what I do is I look back at my Punnett square that I just made, and I can see that there are these two boxes. So 2 out of 4 of the boxes then are going to have a light color phenotype. So 2 of the 4 boxes then are going to be light colored moths because they have the homozygous recessive genotype. One other quick note, so homozygous recessive is the only way then that you can show that recessive phenotype. So again, if the problem says that they have the recessive phenotype, then they have to be homozygous recessive. So here's another problem. Let's say that in seals, the gene for length of whiskers has 2 alleles. The dominant allele codes for whiskers, and the recessive allele codes for short whiskers. What percentage of offspring would be expected to have short whiskers from the cross of 2 long-haired seals, long-whiskered seals? One parent is homozygous dominant, and the other is heterozygous. So again, step number 1, figure out the genotypes of the parents. So for the homozygous dominant parent, they are going to have 2 capital W's to represent the 2 dominant alleles, and the heterozygous parent is going to have 1 capital and 1 recessive allele, so 2 different alleles. Then once I know the genotype of the parents, then I can fill in the top and the side of my Punnett square. And again, note that although the parent does have 2 alleles for each trait, when I put them on my Punnett square, I separate those 2 alleles because they would only put in 1 of the gamete cells. And then top goes down, left goes right, so I fill in, and this is my table. So once I filled in my table, step 3, then I look back at the question, step 4, and answer the question. So the question is asking, what percentage of offspring would be expected to have short whiskers? So if I'm looking at my table, short whiskers would have to be recessive, and the only way that they could show short whiskers as a phenotype is if they had 2 recessive alleles, if they were homozygous recessive. And I don't see that at all. So my answer would be, for short whiskers, 0 out of 4, or 0%. I hope that was helpful to work through at least one of the problems that you will see on the Mendelian Punnett square quiz in this folder. If you have other questions, please let me know, and I can put together another video to hopefully help all. Well, have a wonderful day, and I will talk to you soon.